Hello and welcome to BioChain and Ambergen webinar, everybody. Today's webinar is on unlocking the full potential of biospecimens for multiomic and spatial analysis. This webinar is co-hosted by BioChain and Ambergen. BioChain Institute is a cell bioscientific company, a global leader in high-quality biospecimens, advanced sample preparation products, and services that support translational research, biomarker discovery, and clinical applications. Their ethically sourced and ISO-certified collections empower reproducible data generation across genomic, proteomic, lipidomic, and multiomic platforms in general. Ambergen Inc. is a biotechnology company focused on next-generation spatial biology. Ambergen has developed innovative proteomic and mass spec imaging technologies, culminating in its patented mirrorless photocleavable mass tag imaging system. Today, we will focus on how high-quality biospecimens and integrated workflows enable reproducible, high-impact multiomic and spatial insights. Our presenters will cover an overview of cell bioscientific capabilities with a focus on optimized biospecimen sourcing and preparation, multiomic data collection using mirrorless imaging system, and finally, a case study in demonstration using BioChain's frozen tissues. We will conclude with a 10-minute live Q&A session, so please send in your questions throughout. Before we dive into the presentations, I would like to introduce and welcome our three speakers for this session. We have Dr. Nishantini Durairadru, application scientist and project manager at BioChain Institute. Nisha specializes in oncology-focused biospecimen procurement and maintaining sample integrity. She will discuss how strategic biospecimen sourcing and preparation maximize reproducibility and analytical depth. Next, we will hear from John Gillespie, President and CEO of Ambergen. John has driven Ambergen's expansion into hyper-multiomic spatial imaging solutions. He will share insights into Ambergen's mirrorless imaging platform and its role in enabling high-plex spatial and multiomic data acquisition. And finally, we will hear from Dr. Leonardo Garcia de Torre, bioinformatics lead at Ambergen. Leo develops computational tools that power Ambergen's multi-mass spec imaging technologies and integrates advanced workflows that combine proteomic, lipidomic, and transcriptomic imaging within the same tissue sample. Leo will be presenting a case study using BioChain's frozen lung tumor TMA to demonstrate integrated spatial and multiomic analysis. So with that, welcome all of you, and we will start with Nishantini. Go ahead, Nishantini. Thank you.
Thank you, Rikita, for the kind introduction. Hi, everyone. It's a pleasure to be here, and I'm excited to begin our session titled Unlocking the Power of High-Quality Biospecimens for Spatial Research. I'm Nishantini Durairadru. I'm an application scientist and project manager for biospecimen procurement at BioChain. Today, I will start by highlighting why high-quality biospecimens are foundational to modern cell and molecular research, especially for multiomics and precision medicine. I will briefly introduce cell bioscientific and how BioChain supports researchers with rigorously controlled, clinically annotated human tissues. We will review our comprehensive biospecimen portfolio and then take a focused look at frozen tissue microarrays and their value for advanced workflows. I will also touch on our quality compliance and supply chain standards, including RIN and DIN qualified frozen tissues for spatial applications. Why biospecimen matters? Biospecimen are the ground truth or the foundation of every meaningful discovery in modern biology because they provide the ground truth, the real biological evidence that connects theoretical model to actual human biology. No matter how advanced our technologies become, the accuracy of multiomic and spatial data ultimately depends on the integrity of the tissue we start with. Unlike synthetic or in vitro models, human samples capture the full complexity of disease biology, including the intricate interactions between the tumor microenvironment and the host immune system. These interactions shape therapeutic response, resistance mechanisms, and disease progression in ways that cannot be replicated artificially. High quality biospecimens also enable precise patient stratification. Instead of treating all patients like with one size fits all approach, molecular signatures derived from real tissues allow us to move toward targeted, personalized therapies. In this way, biospecimens bridge the gap between discovery and clinical impact, transforming scientific hypotheses into actionable insights that truly advance precision medicine. It's said that data is only as good as the sample. Unlocking the full potential of biospecimen requires more than just understanding the scientific challenges. It requires an ecosystem capable of addressing them end to end. To consistently deliver high integrity multiomic ready samples, you need the right infrastructure, the right expertise, and the right partners. This is where Cell Bioscientific comes in. Our integrated platform includes Aspix, System Biosciences, and BioChain. Together, these organizations create a comprehensive continuum from sample collection and preservation to biomarker and therapeutic discovery, making Cell Bioscientific a complete ecosystem dedicated to advancing translational research. Our commitment is summarized in our guiding principle, advancing discovery, enhancing life. These are some of our key customers we proudly support across our platform. Our mission is to provide an integrated value chain that advances discovery from bench to bedside. We begin with system biosciences, offering tools like gene targeting and EV systems for early stage disease modeling. And as the research moves from discovery into the preclinical phases, Aspix and BioChain ensures translational continuity through high-quality biospecimen and histology services. The infrastructure carries through to clinical development and final diagnostic commercialization. In essence, Cell Bioscientific is an integrated value chain from basic research through clinical implementation. Let me walk you through how our integrated platform supports end-to-end cell and tissue research. We manage a large repository of human and preclinical biospecimens, including FFPE and frozen tissues. In addition, we also have 300 plus cell lines, creating a unique bridge between cell biology and histopathology. Complementing to this is our extracellular vesicle isolation and gene targeting service platform, offering both viral and non-viral gene delivery tools, as well as EV purification system to support mechanistic studies and biomarker discovery. So at the core is our standardized histopathology workflow. Here, specimens undergo controlled FFP and OCT embedding and array construction, and arrays include tissue microarray and as well as custom cell pellet arrays, and these are delivered as like blocks, sections, or on-slide controls. We also provide immunohistochemistry and in situ hybridization and immunofluorescence services. Then these slides move into digital scanning and advanced analytics, leveraging VisioPharm algorithmic software for downstream analysis. We also provide spatial biology service for 10x and NanoString platforms. We are a 10x certified service provider for VCM and Xenium, and we also offer Chromium single cell. So together, these integrated capabilities form a complete pipeline spanning biospecimen sourcing, processing, molecular analysis, and high-dimensional spatial informatics, all under the Cell Bioscientific platform. Now that we have seen how Cell Bioscientific integrates capabilities across its division, at the center is BioChain, and BioChain ensures specimen consistency, preserving biological signal by clinical relevance across immuno-oncology, translational research, and diagnostics. By providing high-quality FFP and frozen tissues, TMA, blood products, and NGS-characterized sample, we enable seamless integration of spatial and genomic insights to drive reliable biomarker discovery and tumor profiling. Precision medicine doesn't fail in analysis. It fails upstream without the right biospecimen. To understand the breadth of what we can support, let's take a closer look at our biospecimen capabilities. Our biospecimen offerings cover both retrospective and prospective collections, all aligned under strict customer specifications. We provide FFP tissue, frozen tissues, matched pair including like FFP frozen matched pair or like tissue matched with the body fluids. We support with the following body fluids like whole blood, plasma, serum, Buffy coat, PBMCs, bone marrow aspirates, and custom fluid collections like saliva, urine, and CSF. We also provide the patient or the donor metadata, including age, gender, demographic information, pathological information, including clinical grade, stage, TNM, medical history, medication use, treatment status, treatment history, and the custom gene target data if required. So this diversity allows researchers to design multi-layered studies that integrate genomics, transcriptomics, proteomics, metabolomics, spatial biology, all from well-characterized and ethically sourced samples. We procure samples globally following our standard SOPs across all the clinical sites. All our biospecimens are IRB approved with documented informed consent. We strictly adhere to HIPAA compliance and de-identification standards and ensuring no direct PHI is shared. In short, we ensure every biospecimen is ethically sourced, fully traceable, and compliant with global privacy and governance standards. Why customers prefer to work with us? So BioChain is a world leader and we are a one-stop shop for biospecimen sample kits and the extraction kits and the spatial biology services. And we have a strong technical team and it is ready to collaborate with the custom procurement projects and services. We prioritize quality service and fast turnaround time globally, and we have our in-house pathologist who performs QC for every incoming tissues and ensures standard across our deliverables. With 30 plus years of experience and ISO 9001 and 13485 certifications, we are committed to delivering high quality products and services. With that said, having access to sample is only part of the equation. The way those samples are procured and processed determines their true multiomic value. Our procurement and processing workflows are built around consistency and molecular preservation. From rapid stabilization and snap freezing to controlled FFP processing, every step is designed to minimize pre-analytical variability. And this is very critical for multiomic studies where even small deviations in handling can lead to major differences in RNA integrity, protein stability, metabolite retention, and spatial morphology. Next, let's look at the FFP side of a product portfolio, which remains essential for many histology and diagnostic workflow. We offer a wide range of products, including FFPE sections, arrays, panels, blocks, characterized tissue, gene characterized tissues, and all these are performed or processed under standardized condition to ensure consistent morphology and staining quality. So while FFPE is not ideal for all omic layers, it remains indispensable for histopathology workflows. Our goal is to provide FFP materials to complement with the frozen tissues in integrated multiomic studies. Today, I will focus on the frozen tissues. So compared to frozen FFPE, frozen tissues, they preserve RNA and all the metabolite and the architecture with high fidelity, making them essential for omics application studies. Our tissues are collected under strict SOP with controlled ischemia times, rapid snap freezing in liquid nitrogen, and comprehensive metadata capture. This ensures true multiomic readiness. Our frozen product portfolio has sections, arrays, and panels across different tissue types and tumor types. One of the most powerful tools we offer for high throughput screening and assay validation is our frozen tissue platform. BioChain has been a pioneer in frozen tissue technologies, and our frozen tissue arrays are one of the most powerful tools for high throughput screening. These arrays include like tissues from normal donors or like with different tumor tissue types, and also we have arrays for neurodegenerative diseases. Because like each core is preserved in OCT and processed under strict SOPs, these arrays maintain biomolecule integrity, making them ideal for antibody validation and biomarker discovery. So there are like several technical challenges in frozen TMA construction. It is not that easy as like FFPE TMA construction. Frozen TMAs has their own challenges, and the first one being the stringent thermal control. The entire workflow must remain at sub-zero, and even small temperature fluctuation can cause freeze-thaw damage and compromising on the tissue morphology and integrity. The next one is the structural fragility. So without the paraffin support, these frozen tissues are prone to shattering, crumbling, and deforming, and the mechanical pressure may cause core compression. Poor core to block adhesion. This is like because like the frozen tissues are very difficult to punch and make it adhere. It's not easy as the FFPE, and this may lead to sample loss during sectioning or staining. And reduced visualization accuracy. Frozen tissue has a greater opacity, and it makes it harder to precisely target microscopic region and increasing the risk of sampling errors. And ergonomic and mechanical complexity, this because it requires like cryo arraying equipment and precise handling in cold environment. So this makes it's like technically challenging and demanding. Frozen TMAs require strict temperature control, specialized tools, and expert handling to preserve tissue integrity and sampling accuracy. So I'm just zooming on one of the cores to show our tissue quality. Okay. To understand why frozen tissues are so critical for multiomic and spatial biology, it helps to compare them directly with FFPE. While FFPE remains highly valuable for histology and clinical diagnostics, chemical cross-linking and molecular degradation limits its performance across many omic applications. Frozen tissue, in contrast, preserve RNA integrity, maintain the native protein conformation, retain the metabolites, and preserve true spatial morphology, making them the gold standard for high-fidelity transcriptomics, proteomics, and spatial mapping. They are also essential for MALDI mass spectrometry imaging because frozen preservation maintains native molecular distribution within tissue, enabling precise spatial detection of lipids, metabolites, proteins, and drug compounds. For example, Ambergen uses frozen tissue-based MALDI imaging to map biomarkers across multiple layers, enabling fully integrated spatial multiomic analysis. This slide highlights the most critical challenges we see with frozen tissue in multiomic studies and how College of American Pathologists and ISBR aligned best practices directly address them. First is the pre-analytical variability that includes like uncontrolled ischemia time, inconsistent freezing. Even short delays between resection and freezing can impact RNA, protein, and metabolite integrity. The key mitigation here is rapid snap freezing and strict documentation of cold ischemia time. The second is molecular integrity loss. Multiomics is only as good as the weakest layer, and RNA and metabolites are especially sensitive. Consistent ultra-low temperature storage and minimized freeze events are essential with QC metrics like RIN, RNA integrity, and the DV200 and thaw count that are used to confirm the sample fitness. Third, the tumor heterogeneity and purity, and this remains a major biological challenge. Pathologist review and tumor enrichment help ensure the molecular data truly represents the disease. Another constraint is the limited tissue quantity, and frozen tissue often need to support multiple assays, so upfront assay prioritization and low input workflows are very critical. And the storage and handling variability, single-use liquids, continuous temperature monitoring, and validated freezer infrastructure significantly reduce risk over time. Finally, complete and standardized metadata underpins everything. Without accurate pre-analytical and storage metadata, even high-quality molecular data becomes difficult to interpret and reproduce. Overall, CAP and ISPER best practices provide a structural framework to control variability and preserve molecular integrity and ensure frozen tissue is truly fit for purpose. Beyond our catalog offerings, many research programs require tailored solution, and that's where BioChain custom services excel. We provide end-to-end support, including prospective collection, matched sample sets, custom arrays, specialized histology, and molecular workflows. We also offer TMA construction using tissues provided by customers, ensuring that unique samples, the samples could be from rare disease specimens, longitudinal collections, or specialized cohorts. And those samples are precisely arrayed for high quality downstream analysis. Our services are designed to meet the unique needs of discovery team, translational researchers. With that said, high quality data starts with the right tissue. BioChain's frozen TMA preserves RNA, protein, metabolites, and spatial architecture ideal for 3D genome mapping and integrated multiomics. Ambergen leveraged lung tumor TMA for their MALDI mass spectrometric imaging platform, and they were able to compare and visualize lipids, metabolites, proteins, glycans, transcripts, antibody drug conjugates, and lipid conjugated drugs. Together, BioChain's TMA and Ambergen technology provide a robust, high-resolution, and reproducible workflow for spatial multiomic studies. And now I will hand over the session to John, and he will elaborate on Ambergen's technology. And if you have any questions, we can discuss it during the live Q&A. Thank you.
Oh, hey, John. Sorry, I can't really hear you.
Sorry, how about now?
Oh, yeah, perfect.
144 gigabytes. 144 gigabytes is a lot of data. And we're going to show you today how we can extract 144 gigabytes of data from a single fresh frozen tissue microarray slide. And that includes 24,000 biomarker layers. That's a lot of data from a single slide. My name is John Gillespie, and I'm the CEO of Ambergen, and I'm pleased to be here today. Ambergen is a Boston-based company. We are the inventors of a new technology that we're talking about today, mirrorless. Miracle Light is the name of that technology, but it's a platform for highly multiomic data collection from individual tissue slides. At Ambergen, we do two things. One is we're a service provider. Customers provide us tissue blocks, or they provide us slides. We analyze those and we send back reports, including publication-ready images from those samples. We're also an exclusive supplier of our patented specialized mass spec imaging reagents. Our mission is to accelerate drug development through innovative drug-inclusive spatial biology. We can map the distribution of drug. Here on this slide, you can see an image of this is a mouse brain, and we see a gradient here. Red is where this drug is clozapine. The drug is accumulating in the tissue, and blue is where the drug is not locating in the tissue. This is just an example. But what we can do by mapping this drug distribution multiomically is we can use proteins and RNA to highlight drug targets. And then we can see if the drug is co-locating with the target cell population. And we can go beyond that because we're looking at small molecule omics as well. So now we can look at metabolic fingerprints. We can look at lipids and other small molecules to see, is there evidence that a drug is actually interacting, engaging with its target? We're coining the phrase hyper multiomic. Many technologies out there call themselves multiomic, but we can go far beyond just proteins and transcripts. From one tissue section, we can image 3 therapeutic classes. That includes antibody-based drugs, such as antibody-drug conjugates or mAbs. We can look at oligonucleotide-based drugs, like antisense oligonucleotides. And we can also look at small molecule drugs. But together, on the same tissue section, on the same sample, we can look at many different omics. And that includes proteins, RNA, glycans, lipids, and metabolites. It's one tissue section, 3 therapeutic classes, 5 molecular classes on one platform for ease of co-registration. This slide shows a sample, just a sample of the data we can obtain from a single slide. Now, the image here is a little bit overwhelming, but the amount of data that we can get from single tissue sections is a little bit overwhelming. Over here is an image I showed you already, or I showed you something similar to this, but this is a drug distribution image. These images all come from an Alzheimer model mouse brain. After we do small molecule drug imaging, then we do another round of multi-imaging where we look at lipids. I'm showing 3 lipids here, a sulfatide and 2 gangliosides. Our platform is not just multiomic, it's multimodal. So here, what we're doing is doing a fluorescent scan of the same tissue section. We take it out of our MALDI, we wash off the matrix, and we image it in an Olympus fluorescent slide scanner. And here I'm highlighting two markers. Our fluorescent imaging is necessarily low plex, but here, this is MALDI immunohistochemistry, we're necessarily high plex, where we can look at 100+ proteins simultaneously in a single scan. I'm showing 4 of them here. These 4 are very relevant to the sample type, which is an Alzheimer model mouse brain. So we can see NFL, Noonan, GFAP, and amyloid beta 42. This is a Alzheimer model, and you can see some punctate structures here in the cerebral cortex. And these are the amyloid beta amyloid plaques. Here we can also look at RNA. So this is what we call a MALDI in situ hybridization image. And we're looking at 3 biomarkers here, 3 RNA biomarkers. When we're done with collecting all of this data, we can still take our sample and do an H&E scan and get tissue morphological information. The foundation of how we do this is mass spectrometry, and I'm sure many of you are familiar with mass spec. It's a great tool to separate ions based on their mass. Well, here in this cartoon, we have a mixture of molecules. We ionize by firing a laser at a sample that's been covered in a matrix that enhances the ionization. So then we have these charged molecules, and the charged molecules, the lighter ones, make it to a detector first, and the heavier ones take a little bit longer to get through the detector, giving us a spectrum based on mass. And these peaks in the spectrum they can correspond to a specific lipid. We see many lipids, sometimes hundreds in a sample, metabolites, glycans, or a small molecule drug. But generally speaking, without Ambergen, in a mass spec imaging environment, you can't see large molecules. We're going to get back to that in a moment. I mentioned mass spec imaging. What we're doing with mass spec imaging is we're taking this mass spectrometry and we're collecting a mass spectrum at every pixel we scan. This is called a raster technology. The laser scans from port at 00 and so forth through an entire tissue section. Now, the most we're capable of doing here in our lab is 4 million pixels per square centimeter. Realistically, we typically do something in the neighborhood of 250,000 pixels per square centimeter. The way we target those large molecules that won't fly in a mass spec is with an Ambergen mirrorless probe. And this is an example of one type of probe we make. Here we have an antibody. We have a photocleavable biolinker. And then photocleavably linked to the antibody is an engineered peptide mass tag. This mass tag is sort of like a barcode. We have engineered a collection of these. Right now we have 150 of them. So we can look at 150 items, large molecules, in a single scan. We can go a lot higher. We're capable of doing many more. It's just that right now for our customers, this is the most that they've asked us for. This UV lightning bolt is cleaving off the peptide mass tag. You can see there's a difference in size between the size of an antibody and this peptide that I drew here. Now, in reality, the peptide is 1,000 times smaller than the antibody. And that's why the antibody can't fly in a mass spec column. It's just too big and too heavy. So what we're doing is we're functionally converting something big and heavy that won't fly in a MALDI mass spectrometer to something that's light, a small molecule that will fly. We also have the opportunity to put a fluorophore on the antibody. And that's how we can do our multimodal imaging. We can take some of our mass tags, and from the same antibody, we can see a peptide in the MALDI, and we can see a fluorescence signal in the fluorescence slide scanner. In addition, this is how this is an image of what this could look like. This in the middle here is a protein. These are antibodies decorating the protein, and then we have these photocleavable mass tags hanging on the antibodies. What we do is we take a tissue, we stain it with an antibody solution, then we let it incubate, we wash, and then we photocleave, liberating these mass tags. They stay in position, but they're not connected to the antibody anymore. And then we scan in a MALDI, and we can see a spectrum. And different peaks in the spectrum correspond to different antibodies on the tissue slide. And that's how we can reconstruct a spatial two-dimensional image of protein distribution. We can also image RNA, and that's done using oligonucleotide probes. The oligonucleotide probes are very similar to the protein probes that we see, except instead of an antibody as our binding agent, we have an oligonucleotide. We use, now Ambergen didn't, one question I get is our imaging, it seems it's an antibody, and we're reading out where the antibodies are located. Is it immunohistochemistry? Is it like immunohistochemistry? Our technology is immunohistochemistry. It's immunohistochemistry with a mass spec readout. And the same is true for fish. Is our technology similar to fish? It is fish. It's fish fluorescent in situ hybridization with a mass spec readout. And to that end, we didn't reinvent fish. What we did is we partnered with a leader in fish. We partnered with Molecular Instruments. And Molecular Instruments sells a product called HCR Gold. And we have a product that is, we call the Ambergen RNA-Pex Complete, that lets us do fish imaging in a MALDI. This is a typical protocol sequence for us, for a typical sample. It starts with applying matrix, because the M in MALDI stands for matrix-assisted laser decomposition and ionization. We will do a small molecule scan to look for a drug, like that clozapine I showed you earlier. Then we add another layer of matrix to refresh the matrix. And then we'll do another small molecule scan. We'll look at lipids, for example, in this scan. Then we have a workflow where we remove the matrix, we remove the matrix, simultaneously fixing the tissue, and then we wash it to remove all small molecules. So now we have a clean background for the targeted large molecule imaging that we're going to perform. It's at this point that we'll do a fluorescence scan if we're doing multimodal imaging, an immunofluorescence scan. And then we'll apply matrix and do a MALDI scan. So this is a targeted MALDI scan now. I only show one, but we can do proteins and RNA in that single scan. We do it in a method that we call single pot, proteins plus RNA. And then when we're done, we wash off the matrix, and then we can do an H&E section at the end. This is a brief summary of the type of imaging you can get from a workflow like that. On the left, I showed you an earlier picture of a clozapine drug distribution. Here it is again. Here is from the results from that lipid scan, where we're seeing lipids, a couple of gangliosides, a sulfatide. And then here, this is the large molecule scan. And there's 3 proteins shown here, one GFAP, amyloid beta 42, and we're showing 3 transcripts. Now, this is only 10 images. I teased the fact that we were going to get 24,000 images. We'll get to that, but it gets down to the point where Ambergen's technology produces so much data that it transcends imaging. There's only so many data layers you can show on a single slide. And my colleague Leah will get to how we make sense of all that data in a moment. One question I frequently get when I present this information is, can we really scan the same sample multiple times? And well, the answer, of course, is yes. And people have gone much higher than the 3 scans we do. This is a screenshot of a publication from one of the members of the mass spec imaging community, Dr. Erin Seeley, who's at MD Anderson Cancer Center, and she's published her ability to do 8 sequential MALDI scans of the same tissue. Another question I get is, well, does it work? Our technology on a reagent basis has been out there for about 3 years now. From a services perspective, we've been doing services for about 1 year. But our publications in peer-reviewed journals, basically using or referring to our system, have been growing exponentially. This we already have 4 new publications just in 2026. Using this technology, here are some experiments. These are possible experiments. These are all examples of experiments that we've run in our lab for customers. So we start with single omic experiments for people. We can look at proteins, just proteins. We can look at just glycans. We do get more often a call to do just a small molecule drug. We can do small molecule drug imaging. We can also image, I mentioned, three classes of drugs. We can look at oligonucleotide drugs and antibody-based drugs. We can do 2-omic experiments. We can look at proteins plus glycans, proteins plus RNA, lipids plus proteins, any permutation or combination. We've been doing a lot of triomic, 3 different omic layers lately. So drug, proteins, and lipids is the example I showed you just now. But we've also done lipids, proteins, and RNA, or just lipids and proteins. But the most we can do, or the most that people have asked us to do at this point, is 5-omic, pentaomic, where we can see a drug, lipids, proteins, RNA, and glycans, all from the same tissue sample. So using Ambergen's technology that I've gone over here briefly, we can extract an incredible amount of data from an individual tissue sample. When you combine that with the high-quality tissue microarrays from BioChain, it's an opportunity to get this incredible efficiency in terms of how much data you can get from an individual biospecimen. And to go on with this, I'll turn the podium over to my colleague, Dr. Leo Dettori.
All right. Thank you so much, John. Let me start sharing my screen over here. All right. Hello, everybody. I'm very excited to be talking to you today, and I'm going to be doing the final part of our joint presentation, where I'm going to be giving a case study on one of the fresh frozen BioChain tissue microarrays. So this started with a collaboration between the 2 companies that we presented at the European Association for Cancer Research in 2025 in Lisbon. So we analyzed one of their fresh frozen tissue arrays, applied the Ambergen multiomic workflow, and generated this nice poster here that I'm going to be breaking down in the coming slides. So if you're either a reagents customer or a services customer from Ambergen, the heart of our technology is in the lab workflow and the follow-up data analysis workflow. So let's start from the lab. So we start the sample preparation to do a lipidomic scan in this study here. We have our fresh frozen human lung tissue microarray. It's OCT embedded, like Nisha talked about earlier. It has samples including adenocarcinoma, squamous cell carcinoma, as well as some normal samples for control. We start the workflow by taking this light out of the -80 freezer, let it vacuum dry, and come to room temperature for 1 hour in this little chamber here. Once it's ready, this is a very critical step. Every time you're working with OCT embedded samples from mass spec imaging, it's important to try and wash away as much as possible of the OCT embedding material. We do this by doing 3 washes in an ammonium formate buffer with a concentration of 50 millimolar ammonium formate. And this really helps to wash out those residues that could be detected by the mass spec, since the mass spec is a very sensitive tool, and they could definitely interfere with the detection of endogenous molecules. So we try to wash it as much as possible. Once that's done, we put this light back in the vacuum chamber, we light it dry for 1 hour, and then we move on to apply matrix. For detecting lipids, we like to work with a matrix called DAN. Most of these MALDI matrices are organic compounds that we apply on top of the tissue. And basically, they get excited by the laser from the mass spec, and they transfer the energy to the molecules in the tissue, helping them to ionize and fly into the mass spec to be detected. So here in this case, we sublimated DAN matrix, and we took this light to the mass spec. So let's see what some of this data look like. For this experiment, we apply sublimation, we scan under the negative ion mode at a 20 micron per pixel resolution, and we're able to detect 580 lipids. While I cannot show you all 580 in a single image, I can at least show you 4 of them that are color-coded here. For example, we can see this one in gray, which is a more abundant structural one, present in every single core. And it's very important to say that this is a remarkably high number of lipids that we detected. Typically, we detect 200 to 300, but the BioChain samples are such high quality, so well preserved, that we were able to detect a record number of lipids of 580. So moving on to the second part of the workflow, it is our targeted part, where we apply the Ambergen mirrorless technology. So we start by taking this light out of the instrument. We got to wash off the matrix leftovers. We do this usually in cold acetone. Once that's done, we prepare to fix the tissue. We put it in a solution with 5% PFA, and we fix it for 1 hour. Once it's fixed, we can initiate the MALDI in situ hybridization workflow. So like my colleague John talked about earlier, we didn't reinvent in situ hybridization. Instead, we partnered with companies that have fluorescent in situ hybridization technologies, and we switched the fluorescent readout with a mass spec readout. So in this study here, we work with RNAscope. RNAscope has the ability to image 12 transcript probes, and they sell gene-specific probes with a catalog of over 1,000. So we purchased 12 of those, we applied the in situ hybridization workflow, and we used our proprietary Ambergen oligo PCMT tags. So essentially, there's an amplification tree built on top of the transcript, and then we have our tag to be able to detect it. Once that's done, we're able to wash the sample, apply a very mild blocking buffer, and then we can treat it for immunohistochemistry with a panel of antibodies, which are also labeled with the photoclippable linker and the tags. Once that's done, we let it vacuum dry for an hour after doing a few washes to prepare for mass spec detection. After it's all dry, we can photocleave it so we can separate our peptide tags from the detection molecules. This is all done dry so the peptides don't get delocalized. After that, it's time to apply matrix again. Since here we're planning to detect peptides, we work with the CHCA matrix and we spray it onto the tissue because this matrix is optimal for detecting peptides on a MALDI. Once that's done, we're ready to take the sample back to the mass spec and scan it one more time. So let's look at the probes that we use for this study here. For the protein side, we put together an 81-plex protein panel focused on immuno-oncology. So this is the table with the 81 antibody probes that we used. And for RNA, it's very versatile. We can work with any of the probes in the RNAscope catalog. In this case, we had a selection of 12, and we were able to detect all 12. Importantly, Ambergen technology is able to detect far more than 12 probes. It's just that the RNAscope platform is able, at the time, to detect 12. So we continue to partner and work with other in situ hybridization technologies so we can increase the number of transcripts that we can detect on a tissue in a single scan. So going back to the data, we detected proteins and RNA simultaneously, but let's take a look at the transcript data first. So we use the amplification technology here. We detect 12 transcripts, and I'm showing you 3 of those. We can see the transcript for PD1 in red, PDL1 in green, and CD3 in blue. And the colors in white is where we have the overlap of the 3 different colors. We also detected proteins. Here we used our antibody technology to detect 81 proteins. The scan was done this time under the positive ion mode on the mass spec. And while I cannot show you 81 proteins in a single image, we can at least appreciate 4 of them here. We can see the protein CK7 in red, napsin A in green, CK5 in blue, and collagen, a very structural protein represented in gray, which is present in every single core here. So at this stage, we collected a lot of data. So it comes up the question, how do we analyze this data? How can we make sense of this? That's why we developed a specialized data analysis pipeline that I'm going to be talking about next. So this all starts with processing the data, making sure that mass spec data is in an image format and amenable for data analysis. So we do this by looking at the individual spectrum of the data. In this case here, we collected 2 rounds of spectrum, one for the untargeted metabolomics and one for the targeted proteomics and transcriptomics. So the top spectrum here has a diversity of peaks that we automatically pick them, and then we have to tentatively annotate these peaks. To do so, we work with lipid maps, the largest database in the world for lipids. And with this, we are able to tentatively annotate a peak to a corresponding lipid species. For the targeted workflow, it is more straightforward since we engineered these mass tags and we know exactly what what protein or transcript corresponds to each peak. So we developed a Python and Fiji pipeline to be able to process this data and export it as image files. One critical step is to make sure all the different images of the different biomarkers are perfectly aligned. So in this study, we detected 646 multiomic biomarkers, and we have a machine learning assisted pipeline to be able to align the multiple images of the same tissue section to each other. Once it's all processed and aligned, we're ready to go ahead and look at our multiomic images. In this case here, we have a transcript in red, a protein in green, a lipid in blue, and another lipid in gray. So we're ready to start analyzing it. We apply a lot of different tools here in Ambergen to analyze the data, going from statistics to machine learning. But today, to keep it short, I want to show you a machine learning workflow. So going back to the sample had 40 patients. We detected 646 biomarkers, and we apply a k-means clustering analysis, where we identify 5 different clusters to keep it simple. And each one of those clusters, you can look at it as a biomarker expression profile. So we identify 5 unique profiles in the sample. And one way to look at this data is to quantify. We can look at the different clusters, separate them based on disease phenotype, and then we can look at the area of these samples and see what profiles are more prevalent in them. For example, the adenocarcinoma patients, their cores go from A1 all the way through C6, and then we can look at our color-coded map and we quantify it. We see that profile number 3 takes 83% of the area of the adenocarcinoma patients. So this is the more predominant profile for these samples. For the squamous patients, which goes from core C7 to E5, cluster number 1 is the most prevalent one, represented in yellow. And last but not least, for the normal patients, cluster number 5 is the more prevalent one, taking 71% of the total area of these samples. So to make further sense of these profiles, there's several ways to look at them. But one tool that we apply is to look at a ranked list of the top biomarkers that help to differentiate these profiles. So for example, if we look at the squamous patients in cluster number 1, they're highly represented by the protein CK5, which is a known biomarker in the literature for detecting squamous cell carcinoma. On top of that, we can detect a series of other lipids, proteins, and transcripts that are upregulated in the squamous patients when compared to the other ones. So this provides a lot of information in the biomolecular composition of these samples, and it might also help in the future to identify different lipids that might help to phenotype these patients. For example, if we look now at cluster number 3, represented in blue, we see an abundance of the proteins napsin A and CK7, which are biomarkers for adenocarcinoma. And with those, we also see a whole bunch of other lipids and proteins that are upregulated for these patients, but not as upregulated for the other patients. And for the last group, profile number 5, interestingly, we do not see an upregulation of napsin A, CK7, or CK5, which would be expected in normal patients. We also see a very different lipid profile for these normal patients, which could also be useful for understanding how the disease operates and maybe why some of these lipids might be getting suppressed or overexpressed in certain cases. All right, so that's all I had for today on the data analysis. Thank you very much, and we're very grateful to BioChain for inviting us to do this collaborative seminar. Thank you so much, and we're open for questions now.
Thank you. Thank you to our speakers for such wonderful presentations. And we are now open to taking any questions regarding sample quality, workflow optimization, data integration. Please feel free to enter any questions in the chat box. And in the meantime, while we're waiting for these questions, I was just wondering, Leo, is there a standalone software that the users can get access to, or is it a service that you provide for all the integration?
So at the present time, we offer it as a service. So for example, we have a lot of reagents customers that collect a lot of data. Sometimes they don't know exactly how to take this data forward. So we also offer curated data analysis services. So we can get all the mass spec data, we can process it, we work with the customer to test the hypotheses, we find the most appropriate like statistical and machine learning tools, and we deliver a final report.
Got it. Okay. So also I was wondering, Leo, within your workflow, and sorry, I will come back to John and Nishantini as well, but I was just curious, within your workflow, you mentioned that you fixed the tissue with 5% PFA, and I'm just thinking, can you start with fixed frozen tissue instead of fresh frozen?
Yeah, that's a very good question. So we typically try to fix it afterwards because when we're doing the metabolite scanning or, for example, the drug imaging, which is a step that we didn't really do in this study, we want to make sure we disturb the tissue as little as possible. So we don't run into the danger of like delocalizing lipids or delocalizing the drug or potentially degrading something. So that's why we like to start with fixed tissue, and then we fix it kind of halfway through the workflow.
Okay, I do have a question for Nishantini. So Nishantini, you mentioned a lot of great stuff about handling issues and solutions and QC criteria. Is there if you had to point out single most overlooked variable in biospecimen collection or handling that most significantly impacts multiomic data quality?
Thank you, Rikita, for that. So I think if you specifically ask for a single factor that affects the quality, I would say it is the cold ischemia time. So like the clock starts the moment the blood supply is cut off. So even before freezing, the RNA expression or the profile changes in the tissue, and the proteins, they undergo phosphorylation changes and degradations, and there are like metabolite shifts. The architecture or the spatial gradients of the tissues are altered. So by the time the tissue is frozen, there are like the biological artifacts are like introduced into the samples or like embedded in the sample. So I would say the cold ischemia time is the prime factor that's to be considered. And for this, like since we are working with multiple collection sites across the globe, we follow like kind of standardized SOPs to minimize this ischemia time and preserve all the biological or the biomarkers in the tissue.
Understood. Yeah. So tying back to Ambergen's technology, Leo and John, I just have one final question before we wrap up. Do you have any QC criteria in particular before you take on the tissue and process downstream, or is there any consideration that the users need to consider before they send tissue for the service?
So, Rahita, of course, having very high quality samples makes our job very easy. The first phase of any project we do in our lab starts with method development, where we take samples from the customer and we do an assessment on them. Of course, we look for things that are obvious, such as tissue detachment. But we also, for example, if we're doing RNA imaging, we'll do an RNA quality assay at the beginning to see, is the RNA degraded, or in some cases it can be so degraded that we can't read it. So these are examples. We will also do basically dry runs of all the different phases of our imaging workflow in that method development phase, just to make sure that we're seeing high quality lipids or seeing high quality proteins and high quality RNA from our tissue samples. I wouldn't say that there's like one quality metric or even
Right.
three metrics, but it's sort of we're integrating all the quality factors together by making sure that we can get high quality images from tissue samples.
Absolutely, because you're looking into a multitude of metabolites here. So I'm imagining every single metabolite can have its own QC criteria and optimization.
Absolutely. And by the way, a lot of the imaging we do is based on drugs. And drug imaging can be very challenging. We've gotten very good at it here at Ambergen. But we'll start by, for example, taking a drug and spotting just the drug on a metal plate. And then we can basically see what different concentrations of drug solution look like in terms of the intensity that we can see there. And then we'll take it to the next step and we'll look at the drug distribution in the tissue using that as a reference.
Amazing. Sounds good. Yeah, thank you all the speakers, and thank you for all the audience that joined us today. On behalf of BioChain and Ambergen, I would like to wrap up things and thank you all for joining. And a recording will be shared as it's ready to be shared with all the registrants. And we appreciate your time and look forward to continuing the conversation with all of you. Thank you and have a wonderful day.
Thank you very much.
Thank you.
Thanks, everybody. Bye-bye.